This ELISA Tutorial Series is kindly sponsored by Biotech. Please click the link below or in the video description to see what ELISA kits they have to offer. Hi again and welcome to the continuation of our video series on how to perform a sandwich ELISA. So at this point we've finished coding and blocking the ELISA plate, and now it's time to add samples to the plate in order to measure how much of a certain protein there is in those samples. Adding samples to the ELISA plate is probably the most difficult part of performing an ELISA because it can be overwhelming to deal with a large number of samples. To make this process a little easier, I made a 96-well plate sheet which can be downloaded from the Protocol Place website under the Printouts tab. This sheet makes it easy to plan where samples must go on the 96-well plate. Organizing samples and standards. I like to place my standard curves on the far left and far right sides of the plate, and my samples in the middle region. This way, when you wash the samples from one side to the other, one standard gets the first wash and the other gets the last. This will allow you to verify that measurements aren't varying from one side of the plate to the other due to time delays in the various steps. I usually let the last wells of the standard curves be my blanks. This means they only contain reagent dilutant without protein. Blanks are necessary for subtracting out background signal during data analysis. Note that each sample shown here is measured in duplicate. By averaging duplicate measurements, variations in the assay become smaller, and our measurements are more accurate. Arranging duplicate measurements side by side makes pipetting and data analysis a little easier. Once your samples are planned appropriately on paper, arrange them in the same order in your icebox or tube rack. Diluting samples. All ELISA kits are limited in their ability to detect proteins of interest. This means that your samples and protein standards have to be within the working range of the assay in order for you to interpret your data properly. The kit that I use works best for concentrations up to 2 nanograms per milliliter, as can be seen in the manufacturer's instructions. When diluting samples or standards, avoid using black 96-well plates because it's hard to see which wells have sample and makes things a little bit more confusing. Avoid using ELISA or tissue culture plates too, because these can bind to the protein in your samples, making you lose the protein of interest. Instead, use Eppendorf tubes, or better yet, cDNA tubes, which are designed to have low protein absorption. The cDNA tubes allow you to transfer samples to the ELISA plate using a multi-channel pipetter, saving time and effort. Be sure to dilute samples in reagent dilutant instead of water, or PBS, in order to reduce protein adsorption. Once your samples and standards are ready, add 100 microliters of each to the appropriate wells of the ELISA plate. Seal the plate and let it sit for 2 hours at room temperature or overnight in the refrigerator. Thanks for watching.